Okay, so welcome to lab today. We're going to cover the ear. We're also going to look at hearing and also balance. So what we're going to look at are some ear models. And if you were in lab, you would see these different ear models. And we're going to go over these. I hope that you have your checklist out, and I hope that you have your exercise 25 open in your lab book so that you can look at pictures as well. But you can always come back to these PowerPoints as well. But anyway, just a little bit about the ear. So most of us, when we talk about the ear, we're just talking about this part of the ear, which is really just the external part of the ear. That's the part that you can see. But most of the ear is actually inside. And so if you look at that, then in here is where everything takes place. And so we're going to talk about all that. The ear is also connected to the nasal cavity. So there's a tube that runs from the middle part of the ear to the nasal cavity. Now it's called the auditory tube. Some of you might know it as the eustachian tube, but that's named after a person. And names after persons are kind of going away. But what this tube does is it allows air to flow between the nose and the middle ear. So air can flow in and air can flow out. And what that does is it equalizes the pressure on the outside and the pressure in this middle part of the ear in here. And so that prevents this structure here from having too much pressure this way and causing it to flex inward, or too much pressure in the direction and causing it to flex outward. Because if it flexes like this or flexes like this, it doesn't transmit sound as easily. And so we want to keep the pressure in here the same as the pressure out here. And we'll see that a little bit more in a minute. But let's look at it. So when we talk about the ear, we can actually divide the ear up into three parts. So there's the outer ear, that's the part that we see. And then there's this hollow air-filled space in here, and that's the middle ear. And then finally, there's this structure in here where we have lots of neurons, nerve cells, and so on, and that's the inner ear. And so we're going to look at each of these individually. But let's look at the model. So here's our outer ear, the external ear, right in here's the middle ear. There's that tube that connects to your nose. And then over here, these structures are where the nerve cells are. So let's look at the external ear first. So if you look at the external ear, technically it's called the auricle. Auricle just means ear. Sometimes it's called pinna. But if you look at this external ear, it has a very weird shape, has all these little ridges and so on, and you might wonder why it's shaped the way it is. Well, actually, it's shaped that way because it helps funnel air into this tube. And the shape of it actually improves the funneling of the air and the sound into this tube. If you just had a different shaped ear, you get a different amount of sound that's going in. So you might wonder why, like rabbits, if you think about rabbits, they have these big long ears that stick up like this. And so you might wonder why in the world they have these big long ears if it has to do with funneling sound. Well, this part of their ear is not for sound at all. This part of their ear is to get rid of heat. So if you see animals that have these very large ears like this, a lot of them, the way that they get rid of heat, they send lots of blood to this and then heat's radiated away. But this part down here looks very similar to this part right here. Same thing for dog's ear or any other animal's ear. If you look at this part and this part, it's very similar. And again, that's so that sound is funneled into here. And so if you look at it, we have this pinna. The top of the pinna is folded over, and that part where it's folded over is called the helix. We also have a lobe down here, or a lobule. That's at the bottom. And then we have this tube right here, and this tube is what's called the external auditory or acoustic meatus. And it's just a tube through which sound is going to travel to get to this part over here. On the surface of this and all along this little canal here, there are special types of glands. And these are called ceruminous glands. So you might remember this from A and P1, but ceruminous glands are a modified sweat gland. But instead of making sweat, they make earwax. And so earwax gets secreted into this tube. And the earwax, it is a waxy substance. It helps repel water. And so water doesn't stick to the surface or whatever. It moves back out pretty easily. Earwax also has a really nasty taste. If you ever tasted earwax, it's very nasty. And it's thought that it tastes that way because it repels insects. So if you were to get an insect into your ear canal, it is very, very difficult to get out. So if you get something like a roach or a spider or something crawling up in there, it is really difficult to get out, and it can drive you almost insane. So it's much better to repel the insects and keep them from going in there in the first place. So that's our external ear. Let's look at the middle ear. So remember, the middle ear is this air-filled cavity. And the reason there's air in here is because, again, it comes from your nose. It comes from your nasal cavity. So air, just like the air that you breathe in, comes from here into here. And so it's a hollow space. Well, if you look inside this hollow space, you're going to find 3 little bones. This tube, which is called the auditory tube or eustachian tube, pretty soon the name is going to change, and it's going to be called the pharyngotympanic tube. This is a better one because what it does is, pharyngo means throat or the nasal cavity, and tympanic refers to a membrane, which we're going to talk about in just a second right here. It connects those two, and so it's a better name for it. But let's look in here. So here's the external ear, and then here's that middle ear. What divides the two is this. There is a membrane right here. This membrane made out of connective tissue. And it's stretched across even harder connective tissue and bone. It's stretched almost like a drum head. So if you think about a drum head, it's made out of some sort of skin or something like that, and the drum head is stretched. And so if you beat on the drum head with a stick, it's going to vibrate, and that vibration is going to produce a noise. Well, the same thing happens here. When sound comes in here, it comes in and it hits this drum head just like a stick hitting it, and makes the drum head, the tympanic membrane, vibrate. And so that vibration is a result of sound, just like when you hit the drum head with a stick. And so this tympanic membrane, this sometimes called eardrum, is connected to 3 little bones. So there's our tympanic membrane. Here comes the sound in here, and it's going to cause this membrane to vibrate. And when this membrane vibrates, it's attached to a bone, and that bone is called the malleus. And so the malleus, which is right here, is going to vibrate. And then that is attached to another bone called the incus, and that's going to vibrate. And then that's attached to another bone called the stapes, and that's going to vibrate. Well, if we blow those up, they look like this. So here they are for real. They're named for their shape. So someone thought this looked like a hammer. And a hammer is sometimes called a maul. So this is the malleus, little hammer. A hammer beats on a metal structure called an anvil. And because this malleus beats on this, this is called the anvil or the incus. And then if you look at this one, it looks like the bottom end of a stirrup that you'd have on a horse saddle. And the word for stirrup is stapes. So we have these three tiny little bones, the malleus, the incus, and the stapes. So this one is attached to that drum head right here. So if that vibrates, this is going to vibrate. If this vibrates, that's going to vibrate. If that vibrates, this is going to vibrate. And so the sound is going to get transmitted through here. So this is going to vibrate, causing this to vibrate, which is going to cause this to vibrate, which is going to cause this to vibrate. So the sound that you heard changes. It changes from sound to mechanical movement. It changes to movement, vibration. And that's what happens in the middle ear. Any questions about these? These are called the auditory ossicles. Ossicles means bones. So can everybody recognize the malleus, the incus, and the stapes. Any questions? So Nicole says, can you please say that again? My computer froze about the animals. That's not very important. I was just saying that the shape of our ear is to funnel sound into here so it can hit our eardrum. Some animals have very large ears, like a rabbit or a hare or something. And so you might wonder why they have those large ears. You might think, wow, they have much better hearing. But that's not what it is. These large ears are to remove heat. And so they send lots of blood to these ears. Blood is hot, and so the blood, the heat radiates away. So it's a way that they cool themselves. We don't cool ourselves with our ears, and so our ears are much smaller. Does that answer your question, Nicole? So here are those three bones, the malleus, the incus, and the stapes. The malleus, the incus, and the stapes. Let's look at the inner ear. So if you look at the inner ear, it's made out of bone. This is bone right here. It's the temporal bone. If you remember back in AMP1 when you talked about the skull, it's the temporal bone. Well, if you look at this temporal bone, it looks like some sort of little worm or some sort of little animal drilled out holes in here. And the holes, it's like a racetrack or something. The holes are like this. And the shape of it is kind of complicated. And so this shape is called a labyrinth. So in other words, there is a hollow pathway, hollow labyrinth inside the bone. And that's exactly what it's called, the bony labyrinth. Well, not only is there a bony labyrinth, but inside the hollow spaces here, inside this pathway, there's a membrane that runs exactly the same path. It's kind of like putting your hand in a glove. So the glove is sort of like the bony labyrinth, and your hand is sort of like this one, which is called the membranous labyrinth. So the membranous labyrinth is a membrane tube that lines the bony labyrinth. So if we look at it in another view, this is what it looks like if you were to pull it out. So this white here represents bone. So look, it's hollow inside. It's hollow here because it's been drilled out. So that's the bony labyrinth. And then the gray part right here is that membrane that completely lines this tube. And it follows the exact same path. And so that's the membranous labyrinth. We go back to this other picture again. Bony labyrinth. That's the hollow tube pathway through the bone. And then the membranous labyrinth is the tube that follows that same path. Here's another picture of it. There's our bony labyrinth, and there's our membranous labyrinth. Bony labyrinth, membranous labyrinth. And if you just did a cross section through it, there's the bony labyrinth right there, and there's the membranous labyrinth right there. Well, it's not dry in here. There's fluids in here. There are two different fluids. So there's a fluid in this picture, it's green, that's between the bone and the membranous labyrinth. This one, this is called perilymph. Peri means around. So it's around the membranous labyrinth. It's between the membranous labyrinth and the bony labyrinth, perilymph. And perilymph is very similar to the fluid found inside the brain and the spinal cord, the cerebrospinal fluid. So if we look at this picture, this pink part right here, this pink part represents that perilymph. But there's also fluid in here. There's fluid inside here. So if we go back to this picture, there's fluid in here. So this is a different fluid. This is called endolymph. Endo means inside. So it's inside the membranous labyrinth. And the fluid here is different. It's not like cerebrospinal fluid. It's not like that. It's like intracellular fluid. It's like the fluid inside cells. So the two fluids are different from each other. So here, that's where this endolymph is, in here, inside this tube. So here's where we would find perilymph. Here's where we would find perilymph. Here's where we would find perilymph. But here is where we're going to find endolymph. Does that make sense to everyone? So remember, if we do a cross section, it looks like this. So here is perilymph. That's perilymph. And then here is endolymph. All of that is endolymph. Again, perilymph is very similar to cerebrospinal fluid. Endolymph is very similar to intracellular fluid. Any questions? Well, if you look at this inner ear, again, we have this bony labyrinth and we have this membranous labyrinth. But it's also got two shapes to it. So if you look at the two shapes, one of them looks a lot like a snail. Looks like a snail curled up like that. It's helical. This shape is called the cochlea. the cochlea, and this is where hearing takes place. But if you look at this other part, it doesn't look like a snail. It looks like 3 little sort of half circles, or 3 sort of half hula hoops attached to a central space. The central space is called the vestibule. And the little 3 half circles are called semicircular canals. So if we look at it in a different picture, but before we do though, look, there's our little bones right there. There's our malleus, there's our incus, and there's our stapes. But look, here's the same thing here. So there's the tympanic membrane, there's the malleus, the incus, and the stapes, and then here is the inner ear. Again, one part looks like a snail, that's the cochlea. One part is this central space called the vestibule, and then these 3 little half circles are called semicircular canals. You see it better here. Here's the cochlea. Here's the vestibule. And then these are the semicircular canals. There's 3 of them. Does it make sense to everyone? So the cochlea is for hearing. These other things, the vestibule and semicircular canals, are for balance. So our ear is not just for hearing. It's also our organ of balance. It lets us know what our head is doing, which way we're moving, if we're standing upright, and so on. And so if we look at what we've talked about so far, here's our auditory tube over here. Remember that connects to the nasal cavity. Our external ear, you can't see much of it, but there's the external auditory or acoustic meatus. And then here is this tympanic membrane. Again, it's like a drum head. So sometimes it's even called the eardrum because it looks like a drum head. And then we have these 3 bones. So we have the malleus, the incus, and the stapes. And then we have the inner ear with the cochlea, the vestibule, and the semicircular canals. Same thing here. So there's our cochlea. Now part of it's been removed. There's the vestibule. And then here are the semicircular canals. And don't forget, there's both a membranous labyrinth and a bony labyrinth. So here's the bony labyrinth, this white part. Here's the membranous labyrinth, the gray part. Same thing over here. We're going to have the same thing running all the way through. If you take all that out, looks like this. There's our tympanic membrane, our eardrum. There's the 3 little bones. There's the cochlea, part of it's been removed. And then there's the semicircular canals and vestibule. Any questions? Okay, well remember this is for hearing. But hearing, remember, is just like every other sense, it is perceived in the brain. Same thing is true for this. Remember, this is for balance. But balance is also perceived in the brain. And so we're going to have to send signals to the brain in order to interpret what's going on. And so there's a nerve that's going to carry that information. The nerve has two branches to it. One branch comes off the cochlea. The other branch comes off the vestibule. So we have a cochlear branch, we have a vestibular branch, and then together they're going to form the vestibulocochlear nerve. And you should remember this is cranial nerve number 8 from ANP1. So cranial nerve number 8 is going to carry our sense of hearing and our sense of balance. And we can go back and see this nerve in lots of other places. There it is right there. But again, two branches. Same thing here. Two branches. Same thing here, although you can't see the branches. Is this making sense to everyone? Any questions? The vestibulocochlear nerve. Okay, well, let's go ahead and look. Membranous labyrinth is filled with endolymph. Yes, and then between the membranous labyrinth and the bony labyrinth is where we find perilymph. So look at this picture. The white represents the bony labyrinth. The gray represents the membranous labyrinth. The pink, which is between these two, represents the perilymph. Okay, let's go a little more. Well, we already know the cochlea, and we know it looks like a snail. And if you think about a snail, snails are spiral, but there's a tip up at the top of the snail. There's a tip right here. That has a name. It's called the helicotrema. I'm not sure why that is separated, but anyway, the helicotrema. So helix and then extreme. So the extreme end of the helix, the helicotrema. And so we can see the helicotrema in other places too. That would be it right there. Or that would be it right there. Or there it is right there, the helicotrema. Another thing, when you look inside the vestibule, it's separated into two chambers. So even though it's all still membranous labyrinth, they're separated into different spaces. These two spaces, here they are here. One of them is called the utricle, and the other one is called the saccule. So here they are right here, the utricle and the saccule. The utricle is closer to the semicircular canals. The saccule is closer to the cochlea. And that's the easiest way for me, probably is for anybody else to remember which is which. So if we look at it here in another picture, there they are right there. Well, they're not labeled, but this one's closer to the semicircular canals. This one's closer to the cochlea. So this one's the utricle, and this one's the saccule. Same thing here. There they are right there. Utricle, closer to the semicircular canals. Saccule, closer to the cochlea. Any questions about that? There's one other thing we need to pay attention to here. And that is if we look at the semicircular canals, look, they're the same width all the way around until you get right here. So there's an enlarged area on a semicircular canal, and all three of them have one of these enlarged areas. The enlarged areas are called the ampulla. So here's an ampulla. Here's an ampulla. And I can't see the other one, but there's another one as well. So if we go back to another picture, there's the ampulla right there. If we go back to another picture, there's the ampulla right there. There's the ampulla. There's one on all three of these semicircular canals. Any questions? Well, another thing that we can see here, remember this is the Bony Labyrinth. But there are two places where the Bony Labyrinth isn't there. They're called windows. And they're just named for their shape. This one is oval shaped, and so that's what it's called, the oval window. This one though, look at it, it's not oval, it's round. And so it's called the round window. Now we'll talk about what these things are and what they do later on. But there's the oval window, round window. And you're going to be able to see them in all of the models if you turn them over. There's the oval window. There's the round window. The stapes fits right here. So the stapes fits inside this oval window, like that. So usually, we're not going to see the oval window. It's covered up by the stapes. So there's where it's going to be. The stapes is going to cover it up. So the stapes fits right there. And we can see that right here. There's where the oval window is. There's the stapes. Now the round window is not covered up by anything, and you can always see it. Any questions? Okay, well, let's talk about how hearing works. So remember we have this cochlea. So here's our cochlea. And remember it's shaped like a snail. Well, remember we have a bony labyrinth, and inside there we have a membranous labyrinth. And so when you look at it, it looks like this. So out here is the bony labyrinth, and then in here is this membranous labyrinth. It's kind of hard to see when it's all curled up like this. So let's unroll it. So if we unrolled it, it would look like this. So remember right here is the helicotrema. That's the tip of that snail. So if we unroll it, the helicotrema is going to be right there. And then here's bone out here, and here's bone out here. So this is bone out here, the bony labyrinth. And then there's this tunnel in here, this membrane in here. And so here's our membranous labyrinth right here. So there's bone, and then there's this membranous labyrinth, and then over here we have bone. Don't forget we have fluid in these spaces. So there's fluid between the bony labyrinth and the membranous labyrinth. That's called perilymph. Well, look, it goes all the way around. This is also perilymph. And this is also over here, perilymph. It goes all the way around. It's between the bony labyrinth and the membranous labyrinth. That's perilymph. But we also have inside the membranous labyrinth, endolymph. So in here is endolymph. and it fills up this whole space right here. That's endolymph. We already know that. That's nothing new. But these chambers that are created have names. So look, there's the oval window right there. There it is. There's the oval window right there. There it is. And remember, the stapes fits in the oval window. So the oval window is the beginning of this chamber. And this chamber is called the scala vestibuli. And it goes all the way to the helicotrema. But look, it turns around and heads back. And right here is the round window. This chamber is called the scala tympani. So remember, this was bone out here, all the way around bone. So these two chambers have different names, even though they're continuous with each other. And remember, they have perilymph in them because it's between bone and the membrane. But if we look inside the membrane, there's a third chamber in here, and this is called the cochlear duct. So this whole thing is the cochlear duct right here. And it has endolymph in it. So if we take that and we go back up here, it's a little easier to see now. There's the oval window right there. There's the round window right there. So this is the scala vestibuli all the way to the helicotrema. And it's going to turn around and come back. And this is the scala tympani all the way to the round window. And then this is the cochlear duct. Does it make sense? But remember, it's not spread out, it's curled up. And so this is curled up. And so if we look at the cochlea, here it's curled up. But remember, those tubes run all the way through it. So here is one of those tubes. And here is one of those tubes. And here's the other one. But that's what this is too, because it's curled like this. And so when we cut through it, we're going to see multiples of these. Well, in this model, they used color so that you can know that this one is exactly the same as this one, which is the same as this one, which is the same as this one, and so on. This one is the same as this one. Same color. This one, same color. And so, even though when we think of it, we should probably think of it stretched out like this, with this tube in the middle. Remember, it's rolled up like a jelly roll. So when we cut it across, we get something that looks like this. Well, if I were to show that, again, here it is. So this chamber, this chamber, this chamber is the same. Well, all of the green ones are the scala tympani. And all of the pink ones are the scala vestibuli. And all of the blue ones are the cochlear ducts. So, pink, it's got to be the scala vestibuli. Green, it's got to be the scala tympani. Blue, got to be the cochlear duct. Same way all the way around. Is this making sense to everyone? Again, if I took and stretched it out, just unrolled it like unrolling a jelly roll, it would look like this. So here is the scala vestibuli. Here is the scala tympani. And here is the cochlear duct. But when I roll it up, when I make a circle out of it like a snail, and if I cut across it, I'm going to see these different ones. They're different sizes because just like a snail, it gets smaller and smaller and smaller as you get to the top. Well, what if I were to take one of these and blow it way up? It would look like this. So let's look at this. Here's the bone right here. That's the bone, which means this hollow space is the bony labyrinth. But remember, inside this hollow space, we have a membrane. So that's the membranous labyrinth. There it is right there. And so what we're going to wind up with is between the bone and the membranous labyrinth, between the bone and the membranous labyrinth, this is where we're going to have perilymph. Perilymph. But inside here, we're going to have endolymph. So we've got these three chambers that are created, and it looks like this. So if we look at it, there's our scala vestibuli, there's our scala tympani, and there's our cochlear duct. Well, if we blow that up even more and make a model out of it, it would look like this. So look, that looks like this. Well, it's a little bit hard to tell which chamber is which if you just look at it this way. But there's an easy way to do it, and that is to not worry about the chamber, but to learn these membranes. There's three membranes. And these three membranes are going to tell us which chamber it is. So if we blow it up even more, here are those three membranes. This one, which does not look like the other two, is the vestibular membrane. And the chamber that it's next to is the scala vestibuli. That's why it's called that, because it has this vestibular membrane in it. This one, which does not look like the other two, is called the tectorial membrane. And the one with the tectorial membrane is the cochlear duct. And then this one, which does not look like the other two at all, is the basilar membrane. And the basilar membrane is where the scala tympani is. So if we go back one picture and look, there's our vestibular membrane. And there's the scala vestibuli. There's our tectorial membrane. And there's the cochlear duct. There's the basilar membrane. And there's the scala tympani. Does it make sense to everyone? So if I were to ask you to name these chambers, what would A be? What would B be? And what would C be? Even if you didn't have these labels here, you should be able to name it by the way they look. Not only could I ask you that, but I could ask you to name the membrane. So I could ask you what the membranes are called as well. And you should be able to do that. And not only that, but you should also be able to tell me what fluid is located here. What fluid is located here? And what fluid is located here? So Deli says the membranes separate the different chambers. Exactly. So if we go back to our other picture, every single one of these is going to have that. So look, there's our vestibular membrane, there's our basilar membrane, and there's our tectorial membrane. And every one of them has it. There's the basilar membrane, our vestibular membrane. There's the basilar membrane. There's the tectorial membrane. They all have it. Does it make sense? And look, oh, here's that again. And look, here's what, oh, I don't have that picture, but I could show you a real one, and it would look just like this as well. There's one more thing here. So the chambers are all within the ear. Yes, they're all within the inner ear. They're all within the cochlea, which is in the inner ear. This is the cochlea. We haven't talked about those other structures yet. This is just inside the cochlea. Okay, well, remember the cochlea is the organ for hearing. And hearing is going to require neurons. It's going to require nerve cells because it's going to send signals to the brain. Here are those nerve cells right here. Here they are. And if you look here, this organ is the organ of hearing. And it's called the organ of Corti. Well, you see these little yellow things here. Those represent axons. And then look, there are these little weird neurons that have cilia attached to them. Those cilia look a lot like hairs. And so these are called hair cells. So we'll talk about hair cells in just a minute. But this is where they are. And then this is the axon of the hair cell. And the axons, all of the axons, are going to form that cochlear nerve, the cochlear branch. So somehow we're going to create action potentials in these cells here. We're going to create action potentials, and those action potentials are the things that's going to travel to the brain. And then remember, the brain is going to interpret those action potentials. That's how hearing works. So let's talk about hearing. So if you look at this on a PowerPoint, this is a GIF, and it doesn't show up as a GIF in Collaborate, but it still works. So here's what happens when you hear a sound. Let's say it's music. So there's some notes that are being played. And what sound is a vibration in air. So whenever I make a sound with my voice, whenever you hear a drum playing, whenever you hear a guitar or whatever, it creates a vibration in air. And that vibration, that sound, travels as a wave. The height of the wave determines how loud it is. And then that's called amplitude. That determines how loud the sound is. But the waves can be different. They can have different, what's called wavelengths. wavelengths. And all a wavelength is the distance between peaks. So if I look at sound versus this sound, they have different wavelengths. And here's another one with a different wavelength. And if I measure the distance between these peaks, I get what's called the wavelength. And the wavelength determines the pitch, whether it's a high-pitched sound or a low-pitched sound. And the way we measure pitch is by how many of these waves there are per second. That's called frequency. So sound, when you look at it, is different depending on the different sounds. So if I look at this sound, it has a very short wavelength, and these peaks are not very tall. So that sound is a high pitch, soft sound. So it's not very loud, but it's high pitched. But look, if I take that same pitch and I make it look like this, it's the same pitch, but now it's louder. If I do it even more, it's the same pitch, but it's even louder. But look, it doesn't have to have that wavelength. Here's another sound. It looks like this. So if I look at that, that's a medium pitch, soft sound. But look, that's the same pitch. It's just a little bit louder. That's the same pitch, just a little bit louder. And then look, if I take this one and look at it, this is a low pitch sound. And again, the bigger the peaks are, the louder it is. Does that make sense to everyone? Does everybody understand how sound works? And these waves, they can travel through the air, they can travel through liquid, they can travel through solids even, but they still travel as a wave. And so if it's in the air, it's going to travel like this, and it's going to go into your ear. And what's going to happen when it gets into the ear is it's going to strike the eardrum. And the eardrum is going to begin to vibrate at the exact same pitch and same height as what we had here. And then the eardrum is attached to the malleus. It's going to begin to vibrate just like this. And then the incus, and it's going to begin to vibrate. And the stapes, and it's going to begin to vibrate. And what's going to happen is the incus is going to move into and out of the oval window. It's going to push against the oval window. Well, look, this is the size of the tympanic membrane, and this is the size of the stapes. Well, because of this, the sound gets amplified. It's like a little bit like a bicycle. Think about a bicycle. So a bicycle, here's a bicycle, sort of. Looks like this. And right here's where you pedal it. And look, there's a gear here. And then there's a chain, and then there's going to be another gear back here. Well, look, this gear is big. This gear is small. So what's going to happen when you pedal it? For every one time this turns, this may turn 5 times. In other words, we've amplified the force. and that's going to make the bicycle move. Well, look, this is the same way. It has a big gear, if you will. That's the tympanic membrane. And it has a small gear back here, if you will. That's the stapes. And so the sound is going to be amplified. So a little bit of movement here is going to create a lot of movement here. A small motion here is going to create a lot of motion here. So this thing here is only going to cause this to vibrate like this. But when I get over here, even though it's the same pitch, it's going to vibrate like this a lot. So it amplifies the sound. It amplifies the motion, the vibration. That helps us to hear soft sounds because we can amplify it. Does that make sense? Well, remember, here's where the stapes is, right here in this oval window. And so that sound is going to make this thing move back and forth in that window. It's going to push against this sound in the window. Back and forth, push, 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 push. And what's going to happen is, remember, there's fluid in here. And when you push on fluid, it's going to move. And it's going to move with this same wave that we had over here. And the wave is going to travel through here all the way to the tip, and it's going to come back all the way to here. And then what's going to happen is the round window is going to bulge out and absorb the wave so it doesn't bounce backwards. So it looks like this. Here comes our sound. This is going to vibrate. This is going to vibrate. This is going to vibrate. And this is going to vibrate. And it's going to push on the oval window. Right there's the oval window. And here's that perilymph in here. So inside the perilymph, we're going to get this same wave traveling through here. And it's going to travel all the way to here, and it's going to go back. And I'm not drawing it the same, but it should be. And then this is going to move out and absorb the wave. So it only goes in one direction, which is this way. So look, there's our tympanic membrane. Here comes the sound. It's going to make that vibrate. We're going to get vibrate, vibrate. And then here's our oval window right here. And this is going to vibrate. And here's the perilymph. It's going to vibrate and go all the way to the tip. And then come back. And then it's going to be absorbed. Well, remember we have membranes in here. And there's this membrane right here, which is called the basilar membrane. Well, the basilar membrane is different in different regions. It looks like this. So close to the oval window right here, this is a very narrow and stiff membrane. Narrow and stiff. But the further you get away, the closer you get to the helicotrema, the wider it gets and the more floppy it gets. So narrow and stiff at this end, wide and floppy at this end. Well, think about, this is more like, sort of like cardboard. And this is more sort of like paper. So if I stand here and I wave cardboard up in the air and I wave paper in the air, even though I'm waving with the same strength, same arm, the wave for the cardboard is going to look different than the wave for the paper. In other words, if I vibrate this membrane, the vibration here is going to be different than it is here, than it is here, than it is here. Because remember, down here it's narrow and stiff. And the further you go, it gets more and more floppy. So when the sound comes in here and vibrates, some portion of this membrane is going to match that vibration. And it's going to look like this. So look, here are these different waves, and some portion of this membrane is going to match these waves with how floppy it is. Because this one is narrow and stiff, and this end is wide and floppy, each part of this membrane is going to match a different wavelength of sound. And so if you look here, this part of the membrane matches that wavelength. This part of the membrane matches that wavelength, that wavelength, that wavelength, all the way down. And so if we look at it, this first part of the membrane matches high-pitched sounds. Remember, we're going to measure it in frequency. So look at what this frequency is. 8,000. In other words, it's going to go up and down 8,000 times in a second. That's a high frequency sound. But if we look at this part of the membrane, this is what matches it. It's a low pitch, a low frequency sound. Look, it only goes up and down 200 times in 1 second. Not 8,000, 200. But look, a sound came in here. Here comes the sound. There it is, right there. That's the only part of the membrane that's going to match that sound. So this part of the membrane does not vibrate. No vibration. This part of the membrane does not vibrate. No vibration. Only this part is going to vibrate. And look, that particular sound is this frequency. If it were a different sound, so let's say this sound came in your ear. Here it comes. Only this part of the membrane would vibrate, would go up and down. You wouldn't get any vibration here. You wouldn't get any here. You wouldn't get any here. It would just be here. And if it were a different sound, if it were this pitch sound, only that part of the membrane would vibrate. If it were this one, only this part of the membrane would vibrate. Is that making sense to everyone? So kind of like acid-base tests for pH. That's how it compares to me. Well, sort of, I guess. But again, remember this part is narrow and stiff. And this part's wide and floppy. And the further you go this way, the more and more floppy it gets. And so there is a different part of each membrane that compares to each frequency. Over here, it's high frequency. Here in the middle, it's medium frequency. Down here at the far end, it's low frequency. So here's high frequency. Here's medium frequency. And here's low frequency. And if you look, you can see the numbers. Look, 1600. In other words, 1600 times a minute. That's where it matches right there. 800 times a minute. It matches right there. 100 times right there. The very, very end. Look, only 25 or 20 times a minute. Right near the helicotrema. So look at this. Here comes sound. The sound is going to hit the tympanic membrane. It's going to vibrate. It's going to make the malleus vibrate, make the incus vibrate, and make the stapes vibrate in and out of this window. This fluid is going to move. It's going to move with the same wavelength as this one over here. As it moves, it's going to push this membrane up and down. The fluid is. Remember, this is the basilar membrane, and it's wide and flat at this end, or wide and floppy, and it's stiff and narrow at this end. And so, look what it looks like. Here's the membrane. Nothing happened here. This is not moving. Over here, not moving. The only place that's moving is right here. Well, embedded all along this membrane are these hair cells. So look, they're hair cells. And a hair cell looks like this. It has cilia that stick up. The cilia are attached, or they're embedded in this tectorial membrane. There's the tectorial membrane. And when this basilar membrane moves, when it goes up, think about it, if I push this up, the only way that this can go up is if the hair bends over. And if the hair bends over, it sends an action potential to your brain. So this sound, whatever this sound is right here coming in, the only part of this membrane where these hair cells are going to get bent is this part, which matches the pitch. And these ones that are bent are going to send action potentials to your brain. This was not bent. No action potential. This is not bent. No action potential. This is not bent. No action potential. It only happens where we match the vibration. So look, I don't know what this is, but let's say it's music. Well, music is more than one note. It's more than one sound. So it's all coming through here. So look, there's 3 different sounds coming through here, 3 different pitches. Well, if we look, this pitch right here caused that to go up and down, and those cells are going to be bent. They're going to send action potentials. But look, and at the same time, there's also this sound right here, which is causing these to be bent. And those, when they're bent, they're going to send action potentials. And look, there's another one right there where the cells are bent. And they're going to send action potentials. So your brain is going to get action potentials from all three of these. So the brain is going to know there was a high-pitched sound involved in this, there was a medium-pitched sound involved in this, and there was a low-pitched sound involved in this. But there's nothing in between. There's no sound from this and no sound from this. Just here, here, and here. But if I change the music, I change the notes, it's going to be here and here and here. If I change the notes, it's going to be here and here and here and here. And if I change the notes, it's going to be here and here. So as the music plays and as the notes change, different parts of this membrane are going to move up and down, and different hair cells are going to get bent. And so your brain is going to get all of this information, and it's going to come sequentially. And so it's going to hear one note after the next note, after the next note, after the next note, after the next note, after the next one. And what we're going to do is, because different parts of this membrane do that, we're going to be able to interpret this song. Does it make sense? Here's what those look like. So look, nothing much is happening here. Maybe a little bit. It's a soft sound. A loud sound happened there, a loud sound, and so on. And because it matches these different frequencies, we're going to be able to figure out the different pitch of these sounds. And here's what those hair cells look like. So they're bent or not bent. There's our little cilia. That's what sort of what we call hair cells, but they're not hairs, they're cilia. But anyway, they're called hair cells. But the only one that's going to be activated is where the hair cells get bent, and then we're going to get an action potential. Any questions? So once again, look, this is what it looks like. So here's our basilar membrane. Right here, it's narrow and stiff. Way down here, it's wide and floppy. So if I were to take tuning forks, everybody in here hopefully knows what a tuning fork is. Tuning forks, when you hit them, they make a pure sound. One pitch, just one. And so if I hit the tuning fork and put it on your ear, you're going to have just one pitch. One little part of this membrane is going to be affected, no other. So look, if I take this one and I bang it and you hear it, that's what the pitch looks like, and that's the part of the membrane that is deformed. We're going to interpret as a high-pitched sound. But if I take this one and I do the same thing, this is the part of the membrane that's going to vibrate, and we're going to interpret that as a medium-pitched sound. And if I do a different one, I take this one and I let you hear it, and it's going to be that, and it's going to interpret as a low-pitched sound. So right here is where it's narrow and stiff. And right here at the helicotrema is where it's wide and floppy. Remember, it's not straight, this one, it's curled up, but it's exactly the same. So this is this. This is this. And then here are the sounds. So look, these are the frequencies that humans can hear. So about the highest frequency sound a human can hear is 20,000 frequency, 20,000 hertz. And all a hertz is the number of vibrations per second. About the lowest a human can hear is 20 hertz. Well, let me ask you a question. Do you think that there are sounds that are higher than 20,000 hertz? Are there any sounds that are higher? Yeah, there are, but can you hear them? No. So you can go to the store and buy a dog whistle. Everybody know what a dog whistle is? Well, when you blow the dog whistle, you don't hear anything but air being blown out. But what a dog hears is a very high-pitched sound. It's way over here. Because they can hear way much higher pitches than we do. By the same token, do you think that there are sounds that are lower pitched than 20 hertz? There are. But can humans hear them? We cannot. But some animals use very low-pitched sounds to communicate. Whales, elephants, they use these really low-pitched sounds to communicate. Humans don't hear those. But again, there's where the hair cells are, right there. And there's our tectorial membrane. So when the fluid makes this membrane move up and down, these hair cells are going to get bent. Again, that's our organ of Corti. And so that's what it looks like. There are those hair cells. There's the basilar membrane right there. This is where we're going to get ups and downs. And if it gets ups and downs here, this is what's going to get bent. We're going to get an action potential, and the action potential is going to travel down the axon and go to the brain. And the brain will then interpret those sounds. So look, not bent, bent. See, it's straight here, not bent. But look, here comes that wave. That wave of the sound. And that's going to make that be bent. So these are going to send action potentials. These, no action potentials. Here's the picture I was going to show you earlier. This is the real one. So look, there's our vestibular membrane. There's our basilar membrane. There's the temporal membrane. Here's our chambers. And this has perilymph, and this has perilymph. This one has endolymph. Any questions? Okay, so that's one thing that the inner ear does, hearing. But the inner ear is also our organ of balance. And balance is just being able to stand up right. Balance is knowing which way is up, which way is down. Balance is knowing if we're moving or not, and if we are moving, which direction are we moving, and how fast are we moving. That's what this is all about. It's equilibrium. And so when we look at it, it's a different part of the inner ear that does this. It's not the cochlea. It's the vestibule and the semicircular canals. Well, humans and all mammals have two types of balance, two types. One type is called static. Static is not moving, not moving. Static is all about knowing which way is up, what position your head is in. So if you close your eyes right now, you can tell me exactly which way up is. It doesn't matter if you're standing up straight. It doesn't matter if you're laying down on the couch. It doesn't matter if you're doing yoga or something and you're upside down. You know which way up is. So there has to be an organ to help us do that. That organ is found in these two chambers. There's one in the utricle and there's one in the saccule. So that's for static balance. But there's another kind of balance too, and that's called kinetic balance. Kinetic means moving or movement. So when we move, we need to know which direction we're moving, and we need to know how fast we're going. So that's a different kind of balance, and it's in a different part of the ear. It's in the semicircular canals. In these little enlarged areas, remember they're called the ampulla. So we're going to see two different types of balance. So let's look at these two types. So static balance, again, it's just knowing which way is up. Static balance is in the utricle and the saccule, and it uses an organ called the macula. And we'll come back and talk about it some more in a minute. But look, it's got hair cells. Kinetic balance, again, about movement. It's found in the semicircular canals in the ampulla. And that organ is called the Crista. And look, it's got hair cells. So let's look at these hair cells. So look, here are the hair cells. And when you bend a hair cell, what happens is you open mechanical gated channels. We're going to open channels. Hopefully you remember about channels and sodium and potassium and all that stuff from A&P 1. Well, when you open a channel, ions flow. But look, you can bend this hair two different ways. I can bend it this way, or I can bend it this way. Well, when you bend it one way, one type of channel opens. When you bend it the other way, another type of channel opens. So if we bend it this way, we get inhibition. But if we bend it this way, we get excitation. And so look what happens over here. So here are the nerve impulses depending on which way you bend it. You can either speed up the number of nerve impulses or slow down the nerve impulses. You can excite or you can inhibit. And so what it's going to do is we're going to create action potentials depending on which way we bend the hair cells. So let's look at static balance first, and then we'll come back and look at kinetic balance. But let's look at static balance. Remember, it's knowing what position your head is in. The two organs, they're called macula. And there's one in the utricle and there's one in the saccule. And look, they're at 90-degree angles to each other. One of them is parallel to the base of your skull. The other one's perpendicular to the base of your skull. They're in your ear. Let's see if I can find the picture. Here's the saccule, here's the utricle, and they're in here. One of them is shaped like this, one of them is shaped like this. Well, when you look at it, their hair cells is what they are. And here's what those hair cells look like. They have hairs like this, and they're embedded in this sort of jelly-like matrix. So this is a membrane sort of, made out of sort of this jelly stuff. And jelly moves. So there's jelly that can move in here. But in the jelly are little tiny rocks, little tiny crystals. They're literally rocks. They're called otoliths. Oto means ear and lith means rocks. They're ear rocks is what they are. So if somebody tells you you have rocks in your head, you can agree with them because you do. Well, look, these little otoliths are in this jelly-like membrane, and these hair cells are embedded in that. Remember, there's two of these. There's one in the utricle, and there's one in the saccule. One is parallel to the base of the skull. One is perpendicular to the base of the skull. And so they're sitting in, and there they are right here. Well, look, when she's in this position, this is what it looks like. Look, the hair cells are straight up. They're embedded in this little membrane that's got rocks in it. Well, the rocks are pushing down due to gravity, like this. They're being pulled down, straight down. But look what happens when she bends over and changes the position of her head. The head is not upright anymore, it's bent. And look what happens when she bends her head. This gelatinous membrane with these rocks, they're not being pulled straight down anymore, they're being pulled this way. And so this whole thing slides due to gravity. And look what happens to the hair cells. They get bent. And when they get bent, they send out action potentials. And they go to your brain, and your brain knows that your head is now in this position. And then when you go back to this position, look what happens. We're not going to slide down anymore. It's going to move back into its regular position, and now the rocks are being pulled this way due to gravity, and the hair cells are straight up and down. And they're going to stop sending action potentials to the brain. And then your brain is going to interpret that as your head being in this position. Again, it depends on whether the hair cells are bent or straight, whether you get excitation or inhibition. Again, there's two of these. And because there's two of these, one of them, remember, is perpendicular, one is parallel to the base of the skull. There's no position you can put your head in that you don't activate at least one or both of these macula. So again, head straight, head bent. No action potentials, action potentials. Again, there's the little gelatinous membrane, and there's our little rocks, the otoliths. And again, they react to gravity. Any questions? So you should be able to label this. So A, what is A? That's the otoliths. B is the gelatinous membrane. D are these hair cells. Now E, we didn't talk about these, but these are more cells here. They're called support cells. They do not participate in balance. They just support the cells that do. And these are our neurons that are going to send our action potentials to the brain. Same thing here. If we look at this, you should be able to name these different parts. Any questions? So look, this guy is in a roller coaster. There he is right there. Well, look, he's going this direction, and his head is going to get pushed backwards. As soon as his head gets pushed backwards, it's not straight up anymore. It's bent like this. What's going to happen is those little rocks in that gelatinous membrane is going to slide this way, and then those hair cells there are going to be bent, and they're going to send action potentials to the brain. Same thing here. Look, he's standing up straight. These are straight, but he bent over. When he bends over, now the gravity causes these to slide this way. This hair cells get bent. We're going to get action potentials. Same thing here. Head straight up. Look, straight up. These hair cells are not bent. But when you bend your head forward, this slides forward due to gravity, and the hair cells are going to get bent. Make sense to everyone? Any questions? Okay, let's look at the other type of balance. So this is kinetic balance. So remember, kinetic means movement. So we're going to know the direction that we're moving and how fast we're moving. Well, these are different organs. These organs are not found in the utricle or in the saccule. They're found in these semicircular canals. Remember, the semicircular canal has one little area that's fatter than the other part. It's called the ampulla, and that's where they're found. There's 3 semicircular canals, there are 3 ampullae, and so there are 3 of these little organs. Again, they're called the crista. So if you look at a crista, it looks like this. It's got 2 parts to it. It's got one part that looks like a little hat, sort of. And that little hat floats. It's called the cupula. It floats. There's fluid in here, remember, endolymph, so it's floating in here, just like a float on a swimming pool. And then underneath the cupula are hair cells. So if the fluid moves, the little hat moves too. And when the hat moves, the hair cells here get bent. So if we look at it, there's our ampulla. There's the ampulla. There's the cupula, right? The little hat that's floating. And there's our hair cells right there. And so if this fluid moves, that's going to move this way, and they're going to get bent. Well, there are 3 of these. And if you remember geometry, when you talk about geometry, we talk about an x-axis, a y-axis, and a z-axis. And if you know anything about geometry, you know that if you have those 3 points, you can point any point in space anywhere. So there are 3 of these, and they're in those 3 planes. There's one in the x plane, there's one in the y plane, and there's one in the z plane. So there is no way that you can move that you don't affect at least one of these. And because you have 3 points in space, you can figure out where you are no matter what. And again, they have hair cells in here, and depending on which way the hair cell is bent, You either get excitation or inhibition. If you look at those semicircular canals, there are 3 of them. There's an anterior, which means it points toward the front. There's a posterior, which means it points toward the back. And there's a lateral, which means it points toward the side. Every single one of these, all 3 of them, has an ampulla. And inside that ampulla is where that little organ is, the crista. And again, the crista has a hat, the cupula, and then it has hair cells. So, again, there's the ampulla. There's the ampulla. And here's what it looks like. So look, there's our semicircular canal. There's our ampulla. There's our crista. And the little hat, again, is called the cupula, and the little hair cells. And so if this fluid moves this way, this hat is going to move this way too, and these are going to get bent. So when you start moving, the fluid moves with you. And then the hat gets moved along with it. So if you turn like this, that movement is going to cause the fluid to move inside the ampulla. And look, the hat is going to move with it. And look what happens. We're going to bend the hair cells. And when you bend the hair cells, they're going to send action potentials to the brain. And the brain then can interpret the movement you just made. So look, here she is standing, no movement, she's stationary. The fluid is not moving. If the fluid is not moving, the cupula is not moving. And if it's not moving, the hair cells are going to be straight. But the second she turns, fluid is going to move. The cupula is going to move. The hair cells are going to get bent. Any questions? So remember, there are 3 of these, and they're in the X, Y, and Z planes. Well, what if this one is activated? This one's not activated. This one's activated. Then your brain can interpret that and know the direction that you're moving. So, 2 types of balance. So we have static balance. Static balance, remember, is just knowing which way is up, which way your head is pointed. That's this, that's the macula. So what would happen, you think, if this stopped working? Because it can. You can have an inner ear infection. If you get an inner ear infection and these don't work, you don't know which way up is. In other words, you're laying here in bed and you go to get out of bed, but you don't know which way up is. Is it this way? Is it this way? Is it that way? You don't know which way up is. And if you don't know which way up is, you cannot stand. And so when people have an infection like this, they actually have to crawl around on the floor because they cannot stand up. They completely lose this static balance. And then here, remember, this is the other kind of balance. This is kinetic balance. And again, kinetic balance has to do with movement. Which direction are you moving? How fast are you moving? And that is Crista. So if you lose this, if you somehow infect this or it doesn't work anymore, you can't tell if you're moving or if you're stationary. And so you feel like the world is moving around you. You get very, very dizzy and nauseated. You feel like you've been poisoned, actually. And you vomit and all kinds of things. Any questions? Any questions? Okay, well, that's the end of the ear. And so hopefully you'll go back through this, or hopefully you understood it the first time. But later today, the ear quiz will become available. And again, just like you did for the eye, you'll get an announcement, you'll get a notice, and then you can go and take the quiz. But make sure you study before you take the quiz, because remember, you only get one attempt. Any questions? So Nu says yes. So what is your question, Nu? Okay, well, I'm going to go ahead and stop the recording there. Remember, you can always email me for questions.